I wanted to welcome you to this session, which, like I was saying, is a mix of imaging and experimental tooling project demos. In fact, this track in room one is going to be focused on imaging and microscopy. These are very loose categories, as probably you noticed. We try and cluster them together to try and give to try and create like consistent sessions, but we're going to span a pretty broad series of topics. And so I'm super excited to have you all here today. We're going to have Mackenzie present DeepLabCut, and then John on PsychoPy and PsychoJS. Emmanuel is going to present on scikit-image, and we're going to finish with Nicholas and Emmanuel together on Dash.
Thank you so much for having me here. As I mentioned in my quick pitch, I'm from the team for DeepLabCut, and just to give you a sense of our core developers, but of course we have many more on GitHub now. This is myself and Alexander Mathis and Jesse Lahr, who's also here today. So I'm representing this team. And in general, DeepLabCut is a software package for animal pose estimation. And what this really means is that we allow you to track key points or items, pixels of interest, if you will, in animals or other objects across time. And we do this with deep learning. And so today what I wanted to do is to sort of give you a little bit of the background of the project and tell you about some of the things we've been working on. And for the live demo, we actually have a link for you to play with. So if you happen to have any dogs or cats at home, maybe this is the time to bring them out. But nonetheless, let me get started. All right, great. So as you might know, the measuring movements is a really long-standing question, right? We know from exactly, as I mentioned, Edward Muybridge, who had a very simple biological question, namely, did a horse lift all four of its limbs off the ground? And so to do this, he actually had to build this quite elaborate camera trap system on a racetrack, which he called a zoopraxiscope, where he took time-lapse images of the horses and then concatenated them together, and then could answer this question. Yes, of course, when they gallop, they actually do lift all four off the ground. But this is pretty painstaking and laborious work. And of course, cameras and videos have really taken us into a completely different realm. But even back to David Marr, gave us this really insightful vision of knowing that kind of the gestalt or the skeleton of an animal can actually tell you a lot about not only its identity, but of course, over time, how it moves. So you can probably see from these, skeletonized figures that there's probably a giraffe or maybe a gazelle and some other animals. And so in humans or other animals, what's happened is we've been able to track this movement using motion capture systems. So you might be very familiar with this from your favorite animated films like Avatar. And of course, humans were really pretty compliant, right? We get in front of green screens, we'll move around with these markers on ourselves. But this isn't actually not true in laboratory research in the life sciences. So the example on the right is from a really beautiful paper where they put markers on a monkey to understand how the cortex is actually behaving during these 3D meaching movements, but this was really laborious, and it actually took this author about 8 months just to train the monkey to wear the markers. And so this is really, elegant but impractical for large-scale use. And then, of course, things like aquatic animals or mice. I actually run a research laboratory. I can, I'll skip a lot of stories, but they don't like wearing costumes. And so over the years, what happened is we really kept an eye on the computer vision field, and we saw that, human pose estimation was just getting remarkably advanced. And so we thought, okay, how can we imbue some of this really beautiful computer vision work actually into animal pose estimation? I don't have time to go through it today, but we do have different sort of requirements and needs compared to, say, if you're doing pedestrian tracking, you want to know if a person's crossing the road in front of a potentially self-driving car. For life sciences, we want really high accuracy, and we want to be able to tailor this to our specific questions and need. So while these are really incredible algorithms, there was a few things that we, had questions about and wanted to be able to see if we can explore a bit better. So in general, for anyone that's not familiar with deep learning, the idea behind all of these is quite simple. You have an image, you have key points that you want to detect, you have a predictor, which in this case is a deep neural network, and then you get a pose, right? You have to train this predictor, and if you can do some back of the envelope calculations, you'll see you actually need a lot of labeled data to do this. And I really wasn't willing to label tens of thousands of little mouse frames in my laboratory. And so we turned to some other tools by leveraging what's called transfer learning to take pre-trained neural networks and use this in new tasks. And so I'm going to skip over a lot of the science here, but what resulted was effectively DeepLabCut. So what we showed is particularly for this pose estimation problem, that transfer learning is highly effective. So we can take this backbone architecture in this network. This is using TensorFlow and of course, all the amazing tools. It's like I'm a fangirl getting to see many of you that we use in our package. But nonetheless, what you do is you take these images, you label them, and then you train this neural network end to end and fine tune it. And you get something out that's like on the right, which is a mouse reaching for a joystick. And it takes very little user data input. And so that was published just a few years ago. And ever since then, we've tried to make this even more accessible and user-friendly. And so just, as a little aside, if you're interested and don't want to read the full paper, as I said, what was really nice about these tools is that it really required low data input. So on the left, what you're seeing is a graph where the pixel error, you can just think of this as accuracy compared to ground truth data, and then the number of images that you need, and then the ones that are used for training versus test. And you can see even in this example, it only reaches, a few hundred frames to start reaching human level accuracy, or really carefully placing these points on the frame. These are generalizable. They can go to different animals. They can go to multiple animals. So these neural networks are really quite amazing at their job. Okay, and as I had in my background earlier, but I realized a little distracting maybe, this has really been used now in a multitude of settings. So whether you go back to old data from Muybridge, this took about 10 minutes of manual labor to do this and then train a network for 10 minutes. Or we've also worked on cheetahs in the wild for biomechanical questions. And particularly with the help of CZI, we hired a great programmer, Jesse, who's here today as well, and has really helped us push this into new realms like multi-animal tracking and of course all the code maintenance that comes with a larger user base. So we're only 2 years old, but so far we've had about 170,000 downloads and we're used in over 200 universities worldwide. Of course, we have a generous license, so we're even being used in some company applications, which is nice to see as well, and clinical trials. Okay, so we've really taken this software 2.0 approach, as you're all familiar with by now, opening up this code base to make it more accessible to those that don't have programming experience. So we built graphical user interfaces that allow you from creating a project to our own annotation tools, to selecting different types of neural networks, and we're continuing to build and make these state of the art, all the tools to evaluate and refine these networks if needed, and then running video inference and plotting your data and results so you can take it elsewhere. This is just a diagram that I want to say that, of course, we're absolutely built on the open source Python stack. Like I mentioned, we're really using a lot of TensorFlow, Matplotlib, NumPy, Pandas. These are really core workhorses for us. We take these insights from computer vision that we do in our academic research and from others as well. And then we really tried to build it out by utilizing GitHub and this interplay with Collaboratory to let people to explore the data, to Docker containers and larger computing pipeline tools for people like Amazon Web Services or DataJoint, which is used in the neuroscience community specifically. We've also built other neuroscience, maybe specific tools. So Bonsai and Autopilot are two languages that are used to design experiments in laboratories. And the other icon is from OpenEFIS, which is for using collecting brain data. And so DeepLabCut can be used within any of these different ecosystems. And I think I'm most excited about the number of post-pose estimation tools that are really being built on this. So people building example data and or notebooks to use these different packages like scikit-learn, of course, and other downstream tools. Okay, so just as a quick vignette, I wanted to give you a sense of some of the few things that we've been working on. The model zoo, which I'm going to tell you more about. The other one was this multi-animal pose estimation, which you see on the bottom left. The code is already released actually before the publication. And the other one that we recently released is a real-time version of the software package. So you can do this to use in experiments or even at home if you want. COVID kind of sidelined many of us, and here we are. So that'll be out in eLife in the next few days if you're interested, but of course the code is open and available. And we showed that we can build this very nicely into other packages, and it has its own graphical user interface as well. Okay, but I wanted to give you a bit of a demo. So if you'd like to go to this either now or later, What we've been working on is this kind of larger vision of saying everyone is making these incredible tailored networks for their particular tool, and it's fantastic to see that this is being taken up by the community and used in all these contexts, but there are some common things that people might want. And so the idea is to essentially build a zoo of networks that would be more of a plug and play solution for people. So say you have a cat interacting with an iPad at home, perhaps, you wouldn't actually have to label this cat or any of this data for you just to take a video with your cell phone or whatever you like, and then analyze this, because we now have a network that has been trained on many different cats or many different dogs or many other different types of quadrupeds, for example, and a plug and play solution. So also for life sciences and the neuroscience community, we're really trying to build networks that are tailored to common questions that people might be interested in. So if you go to this, you'll find our landing page, which is a very simple Google Collaboratory notebook that allows you to run this. So it's really just as simple as uploading a video directly into Colab, and these are the type of outputs you get. So we have like full dog and full cat. We've had some amazing user contributed tools now, which I'm kind of most excited about in many ways. People are giving back to the project by giving their networks. So say rhesus macaque data. And as you can see from this fun example, we also have a pretty interactive Twitter user base. And so it's been a lot of fun this summer for people trying it, and they just grab a video of their dog, and then, you know, in 5 minutes, there's really no training, and it's a plug and play solution. So this is, of course, maybe not used in research applications, but we hope nonetheless this sort of gets people to be inspired and to use the code base as well and contribute back. Another thing that we've been playing with is actually how to get people to contribute back in different ways. So we have over 50 contributors to the code base now, which is amazing and exciting. The other way is to contribute is through data and helping us build these types of networks. So this has been a really fun, actually sort of kicked off at the CZI meeting in February. So hey, Emma and Nick, we started thinking about how to do community science in the sense of, you know, getting contributors from the outside that want to contribute through a very simple interface. So we turned to making a web app. So you can also go here and contribute if you like. That is wonderful. And two of our talented students helped work on this as well. And so here what you're seeing is data that we've taken with good open source licenses, and then we're pooling this back into these networks for the model zoo. So you'll see lots of things like dogs and cats to make it even more robust, but also research animals like rodents as well. Okay, and so yeah, what we can do then with this data is we get lots of different users. This is a fun kind of computational problem to say what is the best key point that you put forward for training. So it's also fun in an academic setting to look at this type of data. But I think, you know, I really wanted to end on this. I think what is the most important note is that we're really focused on being accessible. And so we've really put a lot of effort into last year about onboarding and user education. So for that, we have a whole series of YouTube videos. So if you're not tired of listening to me today, check out our DeepLabCut YouTube channel. We've done in-person workshops in the past. And one thing that happened to all of us this year is we found ourselves more online. So for that, we actually ended up making a standalone walkthrough course that you can do in your own time. We had a kickoff meeting where we actually had interactive webinars every week, and some of those were recorded. But nonetheless, we kind of take you through the whole pipeline of the user interface to try to onboard people in an interesting way. We've also, you know, been very lucky to have things like the NumPy website feature DeepLabCut, so that's brought in some new contributors, and also recently some other sort of external news sources have gotten maybe, I don't know, interested is not the right word, but written about the project. And so it's been very fun to see this actually taken up in a lot of different venues. And we recently published a paper that kind of is a deeper dive into the kind of the pitfalls and perspectives and the future of what we see for such packages. And with that, yeah, I would like to thank many people, but in particular Chan Zuckerberg, because this is our first support ever for this project that we got from the ground up. And we're extremely excited for what the next year brings, including more of a cookbook style after sort of NumPy and Matplotlib, and of course, a massive amount of code maintenance and some new additions. So with that, I hope there's a few minutes for questions, and I'm delighted to take them.
All right. Round of virtual applause to Mackenzie. Thanks so much. I think many people in this room will be jealous about all the YouTube channel and social gifs you've been posting. It's been really amazing. I've been wondering one thing about this this packaging, this application beyond the lab settings. And I see like I was curious about like going back to the cheetah example of some of the applications in non-lab settings or like a natural backgrounds. And I'm curious about like whether this has been tested in situations where there is a non-stationary background, or if a stationary background is a requirement for this to work, basically.
Fantastic question. No, a stationary background is not required at all. So what's really required is, as long as you can see it, you can track it. And what you end up doing is picking frames that span this diversity. So we actually have all these tools to say, take a video input, do clustering on this to try to pick optimal frames for users to actually label. But no stationary video is required, actually. So it can be you cruising around and it's totally fine. So it has been really fun to see the uptake and more, there's been a huge, even for the sciences of like moving towards more natural behaviors and studying very diverse animals in their ecological niche. And so it's been used for a lot of these different applications. Yeah. I guess I see a few other questions in the chat. I don't know if I should answer them or So maybe the first one, which I think is a really great point, maybe as a comment, but first Stefan, thanks for the cool shout out. It is surprisingly robust, and let me be, there's some really cool challenges, but there's also some really nice robustness here. So kind of as I was just alluding to, it depends on how well you build your initial data set. So of course, we're using these pre-trained networks, which have seen about 1.2 million images ahead of time. So they're really imbued with natural scene statistics. So that already gives it quite a large advantage for this really problem in AI, which is this out of domain robustness, right? Like how do you generalize to these new contexts? So that's actually one of the things that we work on in the laboratory, is like building small data sets and seeing how well they generalize. And that's really pushed like the architecture and development in our hands and data augmentation methods. So it's certainly not a solved problem by many means, but I think it's relatively solved in the sense that as long as people are generating data sets that capture the diversity of what they're doing, it works quite remarkably well. So it was even shocking to me early on. So I'm with you.
Nicholas?
I guess I have two questions for the for the sort of crowdsourced models and data. How do you approach sort of quality assurance on that? And then maybe related, when someone's contributing a model to the model zoo, is it like just the weights, or is it the trained keyframes? Like, are you allowed to take two models for the same kind of monkey and like merge them, or like what happens if they disagree, or do people submit bad frames? Like, how do you approach that bundle of questions?
Yeah, no, that's a fantastic question. So right now, the way that we've done for the external contributions is we have kind of an internal pipeline for doing quality control. So we check and see what they labeled, but we are thinking of new ways to actually outsource this. Because one thing for some of these animals, like having an expert eye for like understanding exactly where the underlying joints are can be really helpful. So there are people that are willing to kind of, with an expert view, like, for example, we take some of the iNaturalist images, and that's a really amazing example of crowdsourcing. But also building an app where people could go back in and correct is another way that we're trying to roll that out in the future. But right now, it's been kind of an internal pipeline, which we all can know is maybe not the most efficient. Your second question is great. So people can contribute the models themselves or the data or a combination thereof. What happens is if they contribute a model, what we ask is that hopefully in the future, if they'd be willing to add in more data and train it, or if they don't want to share the data publicly, we're happy to maintain that on the back end and kind of put that through our automated pipeline of merging these data sets. Because you're absolutely right. That's really the key to make it even more robust, is just these really diverse data sets. So that's exactly what we're trying to do. So I hope that answered your question.
Absolutely.
Cool. Yeah, and great to see you, by the way. Yeah.
So I was just interested in what issues did you encounter when you were building a desktop GUIs for DeepLabCut? Issues like how do you do platform-dependent testing? How do you package stuff? How do you do these very platform-specific things that are targeted at the end users who may not be that experienced in Conda and in all that programming-like stuff?
Yes, yeah, that is an absolutely great point. So the first part for testing, you're right. There's no, and or maybe someone knows and I'm unaware, so I'd love more advice on this, but kind of the continuous integration style of testing for GUIs is hard across different platforms, right? In many ways. Yeah, exactly. So really what that ends up being is when we make a major release of the GUI, Jesse, Alex, and I get on a bunch of different operating systems. Oh, sorry, my office lights are turning off here. And test, sorry, it's a little late in Geneva, so good evening. So yeah, that has been a bit of a challenge in the sense that it's a labor of love to test GUIs and to make that work in many ways. We also early on made some decisions based on what we thought would be the most like cross-platform type of application. But one thing actually that we're doing in the coming year with the CCI support is actually moving to Napari, because we found that to be quite well maintained and much easier to go across platform. So, working on packages that have strong support, I think is really important. But you had a second part to your question, and I'm really sorry, I'm forgetting now what you asked.
Yeah, I think that's it. Maybe
okay? Yeah.
Maybe something about user experience. Like, how would you ensure that like widgets that you're using are actually understood by users and that kind of stuff?
Yeah, that's a great point. So I think it's really been two ways, maybe three is so for any of the GUIs, actually make tutorials about how to use them, and any of like the kind of buttons or kind of weird things that they can get themselves into. We also try to have like help buttons for every kind of main page. So we laid it out in a way that it sort of mimics the pipeline of going through like creating a project, data curation, data labeling, like training networks, and they can stop at any point and reload on that tab effectively. And so I think just the user outreach and being able to talk to them both on like image form and getter and these things have been helpful actually for me to see a lot of limitations and where we can make it better over time. But I agree, there's still, so we deploy it as a conda file, and so we provide that, but that there is like you're right, there's this like basic thing that they have to know, like what terminal is. Like at one point, all of us didn't know what terminal was, right? And so like getting some onboarding for that has also been just part of it to like make this not scary, and like also updating. I think a lot of life scientists and maybe John could talk to this point too. They don't want to always update their software because they're afraid of breaking something that's working, even though we're trying to make it better and better. So I think like showing them that it's always backwards compatible, like engaging with them on the forums like Twitter and being like, try it, you can make a new conda environment, you don't have to throw away anything. That has been really the best way. But I'm very excited to hear if other people, I mean, that's part of this for me too, is like learning from you guys if you have other tips and tricks. So
I mean, we have the same problems.
Yeah.
I guess exactly the same problems. Yeah.
Yeah, exactly. Well, we can have we can have a support group for each other if you need on the side. So yeah.
Yay, ruining the unstructured time session.
Yes.
Fantastic. Thanks so much, Mackenzie. Thanks to everybody who asked questions. Our next presentation is by John, PsychoPy and PsychoJS.
Cool. So this project was about PsychoPy, which is one piece of software that we're aiming to do lots of things. So the idea was that with a single software package, we could have something that was precise enough for psychophysics, easy enough for teaching, and by that I mean first-year undergraduates in psychology who aren't usually a fan of computing, and flexible enough for everything else. So people who are doing, a professor in neuroscience doing EEG, or now someone running experiments online. So we were trying to do lots of different things, and we've been doing this now for the project was begun in 2002 on SourceForge. So we've been around for a while, but it's been a gradual development from as those things have expanded. To achieve those different goals, we've ended up with a sort of quite a multi-threaded approach. So we've got an application that I'll show you in a minute that has a graphical user interface. That's what allows us to be accessible for teaching level, for undergraduates and whatnot. Then it will output scripts in either a PsychoPy Python library. So all of that's open source software, and this is how the project actually began, was with the library. And the key to the library is that it's an OpenGL-based Python library that uses hardware-accelerated graphics for us to present our stimuli precisely in time. And then more recently, we've added this JavaScript library. So Alan Biccio and some others have ported the Python library to JavaScript so that it could run in the browser as well. Currently using Pixie, but we're soon going to move to WebGL. Again, aiming to use the hardware-accelerated graphics that you can now use in a browser. And alongside that, we've been developing Pavlovia.org, which is our software as a service hosting service that has sprung out of this. So I'll just talk you through each of those parts. This is where I'm going to jump to just showing the applications for real. So this is the sort of oh no, there's the problem. I lose my mouse when I do that. Right, I will have to go and quit that first then, that's fine. So this is our graphical builder interface. And this was created kind of thinking that students needed a way to create experiments because they couldn't write code. And that was the way it was framed. But increasingly, actually, this is what I recommend people use even if they can write code. You know, I'm now telling people I would use Builder to create an experiment. So unless you're a better programmer than me, I recommend you use Builder. And that's not to say that I'm a good programmer, but compared to most undergraduates, certainly I am. So it's turned out to be a really flexible interface that allows us precise timing of, say, one trial. So here's the nav-on task where we present a fixation point and a stimulus. We quickly cover it with a mask. Participants can respond from the moment the stimulus comes on until this point in time. And, you know, people can look at this and quickly understand how their experiment works. They can also see how different parts of their experiment fit together. So we've got some trials with some feedback in a loop. Then we've got some more instructions and then some more trials without feedback, etc. So it's a reasonably flexible way to view things. And from that, we can then either compile a Python script, which comes up in our code editor, or we can go and compile now a JavaScript. And again, we can then go and see that in our own editor. That's, you know, for someone who is learning to code in either of these languages, they can now go and explore how this experiment works and see the underlying guts of the thing. They can go and look at the code for the end of their trial. It's reasonably commented, and they can look through and see how it all works. Some people will still be writing their code from scratch, and that's perfectly fine as well. So those are sort of, that's how we get the ease of use and maximum flexibility, maximum power. Anything that Python can do, and pretty much anything that JavaScript can do, that means we can do it as well from any of these scripts. You could go and hack that script and run it from here instead. We kind of recommend that people don't do that, because once you're in the code, it's a one-way street. You can't then go back. I can't change this script here and then expect it to appear back in the graphical interface, because obviously people can insert any arbitrary code there. But what you can do is insert code back here. So I'm going to just show you in the feedback component, in the feedback routine. We've got here some, oh, let's go up in the other window. We've got here some code that, again, it can be in either Python or JavaScript. And actually, Alan has done a wonderful job of allowing us to auto-translate. So I can't write JavaScript. I don't know anything about JavaScript. But I can write else message equals, oops, got that wrong. And it auto-translates the Python into JavaScript for the code component. So this wouldn't work for, we advertised people, we told people that this was possible, and now they're like, I want to convert my entire experiment. Why can't I turn everything into JavaScript? So unfortunately, we've set people's aim to, but it does do amazing things. So first of all, you could say, I don't know, print hello. It knows that that's a console log, but you can even do things like def my func. Print this. It creates a function with the name of my function. So we can do all sorts of things just with auto-translating code snippets into JavaScript. That has allowed a lot of people who haven't had a lot of JavaScript experience to be able to get their experiments running online, which is a huge thing. To be able to run their study online, it's not just a matter of generating the scripts, of course. They also need to then go and upload it somewhere. So that was what Pavlovia was originally created with that goal in mind. But actually, and that's what these buttons are for, so we can push, so we can play our experiment online, we can sync it online, we can explore. So we can now go and find, I don't know, let's search for memory. Oops. Oops, have I not gone on? Maybe we can't. Oh, I've got a dialog box come up, that's why. Maybe I haven't got a connection to this. Never do a live demo, never. For some reason, oh, now we're there. So we could go and search for tasks that are something to do with memory, go-no tasks and false memory tasks. These are tasks that people have put online on our Pavlovia repository. Now let's just go to that pavlovia.org. They can go and explore experiments here. So let's say core C blocks tasks. That's just jumped into my head because I saw that there. We can go and find, has anyone written the core C blocks task? Oh yeah, a bunch of people have got versions of core C blocks tasks. Underneath this repository is actually using GitLab. So you guys are all used to GitHub. GitLab is the open source version of that. And that means they've got all of the flexibility. All of our projects that they're uploading, their experiments, are using Git. When we synchronize, it's doing a Git push and pull. They can fork experiments, they can star experiments. Data are just being pushed into here as CSV files on the whole, although there are other options as well. So what they've actually ended up with is a really simple, either a simple interface using just Pavlovia or a fully fledged repository like GitHub. And we think that's actually turned out to be really useful as well, because if I were to, I mean, they could have uploaded their code to GitHub, but if they upload the code to GitHub and I searched GitHub for memory, I'm not going to find a neuroscience experiment on memory, okay? So this has allowed people a place where they can upload their code and a really useful sort of repository for relevant experiments in their field. So that's actually been a really helpful step as well. Now, I'm going to go back to using some of these slides. Let's do that. Yeah, we've got these various interfaces, and because all of that was, oh no, I will just go back to look at this. Because all of that is hardware accelerated graphics, the other thing that it allows is for us to do dynamic stimulation, dynamic stimuli. So we can change our stimuli on the fly every screen refresh. It allows you to create, brain-computer interfaces, for instance, or just run more interesting experiments than psychologists used to run. So I just need to move this to be in a window, not full screen. Let's just run this here. So it allows us, for instance, to create experiments that are not just basic waiting for a stimulus and pressing a key. Again, oh, here we go. We can do things like have live stimuli that are moving. So this is moving around with the mouse. I'm searching for the football. I find the football. This is probably actually going to be quite jerky for you across Zoom, but found it straight away. On a regular desktop install, this would be a live interaction, and all we were actually doing was moving a mask based on where the mouse was positioned. But because we can do that on every frame, and we didn't need any code for this, all we needed was to create the trial in such a way that this mask was altering its position on every frame, set every frame based on mouse pos. So it allows us to create quite rich dynamic experiments very easily. Okay, I will go back to the slides if I've got a few moments. Now, for many years, I've got halfway through writing that slide. For many years, we had volunteers, and then Funding Why Welcome to Us, and then CZI. And I've obviously failed to finish that slide. But this is allowing us to move into a new domain for us. So we are now, we've created our own company called Open Science Tools Limited, which receives income from that Pavlovia website. So Pavlovia is not open source. We provide software as a service where we've now got over 100 site licenses. So an institution would pay £1,500 per year. We've deliberately kept that extremely low. The sort of commercial alternatives would be £10,000 a year for a site license. So we've kept that very, very low, which is probably why we've got so many site licenses now. And that's generating revenue for us to now actually employ our own staff to do this long term. So CZI has actually filled for us a very critical gap that would have been there in our funding. But now, partly because COVID actually did help us with Pavlovia. It's one of those places that suddenly one of our USPs is that you can run your study online or in person. And suddenly with COVID, that was actually really useful. But we're now actually running workshops and consultancy as well. And we're actually successfully, seemingly, transitioning into becoming a company that still makes free open source software. So living the dream. It's pretty exciting. Yeah, we're getting more and more professional. I'm not going to say much more about that. So increasingly now we've got this team of staff. So we've got Todd and Thomas, who are both funded by the CZI grant. And we've got Alan, soon Matthew, he's joining us in January, and Sotiri and Becca, all funded now by the company. And so it's going to be great fun going forward. So if people want to talk about that side of things as well, rather than just the software side, I'm happy to chat with people about how we're making that transition and keeping free, but also, or as far as possible free, micropayments, and managing to transfer into professional anyway. Otherwise, thanks to lots of our contributors. We've still got contributors coming. One of the things that we were wondering about is when we had professional staff, would that mean people stopped contributing on GitHub? And that hasn't been the case. So we're hoping that we can make this fully work as a dual route, free and funded system. Okay, any questions?
Hi, I'm Eric Ziegler. So I'm working with the OS Viewer project, and I just think that it's really interesting that you have a business model like that. And I was wondering, do people tell you how they're paying your site licenses? Are they paying it like one-off out of their own grants, or is it that multiple people in the lab are contributing to pay for it?
Yeah, huge variety. In fact, so I didn't mention there's also participant payments. So a single lab could just buy credits to run individual participants in their study. Those are 20 pence each. And some labs would do that. In fact, as well, an undergraduate will put £5 on the server in order to run their subjects. Site licenses, sometimes at that price, it's cheap enough that yes, some labs have a grant. Sometimes they'll club together with a few labs and they'll buy a grant. Sometimes it is a departmental decision that they're going to buy it for teaching purposes, say, for the whole department. So we've seen quite a range of different options there, and because we're deliberately making it cheap, that's the idea. I mean, basically, what all of us as open source developers have as an advantage is we've got these tons and tons of users. So you only need a relatively small percentage of those users. When you've got 10,000, you only need a small percentage to pay and to pay quite a small amount, and you generate a reasonable, a usable income that you can pay further development with. So that's kind of been our model. Whereas the commercial people have got maybe 20 staff from day one with no customers, and they're starting to build up from there. They therefore got to charge a high price, and therefore it's hard to generate more custom. So I think open source developers, we should leverage this fact that we've got, and our users are not just users, they're fans, right? People will sing from the rooftops about their open source packages that they're using. So we're looking at how do we leverage that and still not charge more than pennies, but make that work for other people.
You should absolutely join
really
the audience building session with how to build your fan base suggestions. I love the idea of this sustainability model. And next up, we have Emmanuel presenting on scikit-image.
Hi everyone, my name is Emma and I'm representing scikit-image. Scikit-image is the image processing toolbox for scientific Python. We've been funded by CZI for EOSS round 1, and the people who have been funded for this work on scikit-image are Marianne Korvelek and myself. But the work I'm going to present today received contributions from many more people. Of course, the scikit-image core team and all the people, all the volunteers who contribute to the project. In this EOSS grant on interactive image processing, the other part of the grant was developing interactive image processing with a Plotly Dash, and this will be the subject of the next talk. Before I move on to describing what we've been doing on scikit-image, I like to say that before this grant started, I had been contributing to open source software as a volunteer for almost 10 years. And for me, it was really a great opportunity to be funded, to be paid really for the first time of my life for working on open source software. So thank you, CZI, for allowing this. Scikit-image is the image processing toolkit for Python. It's a go-to library if you have some image processing to do and NumPy arrays as images. So with scikit-image, your image data are NumPy arrays, which are the common objects when you do numerical computing. And then scikit-image gives you a collection of functions which you call on your NumPy arrays and which you can call in a pipeline way to apply different operations on your image data and to extract information from them, like, for example, filters, functions to measure things in your images, and so on and so forth. scikit-image is for scientific imaging, and therefore it caters not only for 2D images, but also for a variety of 3D imaging modalities, like the images people get in CT or MRI. We, in some functions, we even accept n-dimensional images. And scikit-image is a community project, and for a long time, it was not funded by a specific organization, and we try to have as many users as possible, including people who are just starting programming, students who are learning life sciences, physics, and who want to process their images. And therefore, scikit-image has a strong focus on simplicity. It's really one of our core values and part of our mission. And as for simplicity, we have this very simple functional API where really almost all operations are just functions applied to images. We don't have fancy classes or complex objects. And in terms of our code, we are based on NumPy and Cython code, which is quite easy to maintain compared to, for example, C++. I can tell you that it's already hard enough to maintain with just NumPy and Cython if you try to be compatible with most architectures. In order to have a smooth learning curve and to attract as many users as possible, we also focus on pedagogical example-based documentation with a lot of examples in our documentation. In terms of features, functions available with scikit-image, we have on one hand, both really simple utility functions. For example, when you want just to rescale an image to change its linear size, things that could be done with a few lines of NumPy. Sometimes we just encapsulate them so that people can call a one-liner of a scikit-image function. And on the other hand, we also include really more advanced algorithms, like, for example, this segmentation algorithm, which relies on machine learning and has quite a complex architecture within the function, but which is hidden, the complexity is hidden to users. So that, once again, it's just a one-liner call to one scikit-image function, which of course can be chained together in a pipeline way in order to build an image processing workflow. Scikit-image is application agnostic, but it's used mostly in science and in various kinds of scientific applications. If you look at the citations of the scikit-image paper, which has been cited more than 1,000 times, you'll find papers working on medical imaging, on cell biology, astronomy, material science, and so on and so forth. So quite a large variety of applications. And if you look at the community in general, it's quite large. We're really one of the packages which are part of the base of the core of the scientific Python ecosystem. And for example, the statistics of visits in our documentation website during the last 3 months, we had more than 300,000 unique visitors, so quite a thriving community. Then fortunately, we also have a lot of contributors. For each release, we usually have more than 50 people who contribute to the release. In order to mentor these contributors and to help the contributions go through the finish line and be merged into scikit-image, we have the core team, which is not 300,000 people, not 50 people, but around 10 active people. So we have this funnel of, which is classical in scientific Python and in software in general, of a large number of users, quite a large number of contributors, but much smaller, and then again, a smaller number of maintainers. As a result, even if we try to be as reactive as possible, we have a backlog of pull requests on the GitHub of scikit-image, and this is why it was so important to get funding for the maintenance of scikit-image. After this brief introduction to scikit-image, during this talk, I'll focus on different aspects, maintenance and community. Then I'll move on to what we've been doing with documentation and finish with a few words about new features. In terms of maintenance, here are a few figures to give you an idea of the daily life of scikit-image in 2020. More than 400 pull requests merged with a trailing backlog from this 400 pull requests of 40 pull requests. So it's 10%. You can consider it's either not so bad because it's only 10%, or that it's 40 contributions which are still missing. So it's something we are still trying to improve. We had 2 releases, 0.17 and an ongoing 0.18. You can already download, pip install the release candidate and tell us if it's breaking everything for you. We hope not. Each of these releases had more than 50 contributors, something which really happy about. And one point I'd like to highlight about maintenance is that this year has been quite a continuous integration marathon with quite a lot of changes in our CI matrix, in particular moving from Travis to GitHub Actions. One thing which has been really useful is adding doc artifacts to our CI matrix. When you have a pull request, you can see the doc which is generated inside the pull requests thanks to Circle CI. And this is very useful if you want to test the new gallery example that a user is contributor is providing, so that you don't have to download their branch, and it really reduces the friction for reviewing pull requests. In terms of community and outreach, in order to grow this community of contributors, we had 2 sprints, one at the PsychoPy conference and one at PsychoPy Japan. Of course, the 2 sprints were fully remote. And of course, we missed the good atmosphere of in-person sprints. But as you can see from this picture, remote sprints also have some advantages. For example, being able to contribute with a baby or a toddler on your lap. As you can see from this picture, where we had 3 babies during the scikit-image sprint. So it's also a way maybe to reach other people who could not have participated to an in-person sprint. And we gave talks at different places, both Python conferences, also local meetups, applied mathematics conferences, trying to spread the word about scikit-image. We also tried some improvements to the organization of the core team. So most people in the core team are volunteers, and therefore for them it's often hard to be aware of what's happening, what's going on in scikit-image. Like you're at some point you have a lot of time to contribute to scikit-image, and then you're busy with your day job, and it's hard to catch up. Catching up is really a challenge for core developers. And so something we set in place was a rotating community duty, where every week you have one person who is in charge of answering issues and triaging pull requests, but also issuing sort of newsletters for contributors and core members saying, hey, during this week, here are the new pull requests, but also there was activity on this older pull request. Maybe some attention is needed for the specific pull request and so on. So it's a way of making catching up easier for contributors and core members. So this has worked out quite well for us, and I hope we can keep on having energy on this. We also had more frequent developers meeting. And one thing I'm really happy about is that thanks to CSDI funding, I was able to mentor Marianne Korvelek for contributing more to scikit-image, in particular contributing documentation examples. And recently she became a core member of scikit-image, which is great. Marianne is a great addition to the team, and in particular, so during quite many years, I've been the only woman on the scikit-image core team. And so the core team has always been a very friendly and safe place where you can learn a lot of things and make friends. But it's really great as a woman when you have a second woman joining the core team, I can tell you. Let's move on now to documentation. So in terms of documentation, one first thing to do was to measure the documentation analytics to see if people were using our documentation. And for this, we chose to use plausible.io, which is an alternative to solutions which are less respectful of users' data, such as Google Analytics. Plausible is completely GDPR compliant, for example. And so this was a way for us, okay, to be happy about the number of visitors, but also to see which are our top pages, like the install page is our top page, visited page. This was quite a surprise for us, and we rewrote it quite extensively. When you take a look at the bounce rate, it's a bit sad because then you see that it's quite high, and it means that a lot of people go to the documentation website, but probably they don't find what they are looking for. And in order to make sure that the documentation pages are not dead ends, we added some little things to documentation pages, in particular in our gallery of examples. So we added links to Binder notebooks if you want to run one example live. We also added links to other gallery examples. Hey, maybe this example was not what you are looking for. See also these other examples. And last but not least, we added a link on every example page to the users forum, the image SC forum, so that if people still have questions, they can ask them on the forum. About the documentation, also, we are aware that a lot of scikit-image images, like data examples, are on general purpose images, such as images of cats or astronauts. But it might be hard for life scientists to relate to the applications. And therefore, we added examples which were specific to life sciences. And this was in particular the work of Marianne. And for this, we needed to include larger data sets into the package. And we used the Pooch package, which has some clever fetching mechanism in order to do this. So last but not least, about documentation, we also wanted to make the documentation more interactive so that when someone lands on a documentation page, like this one, can you see? Yes, the documentation page. So they can see the code, but they can also interact more with the image and in particular explore the output of the example. They can zoom, they can pan, and for this, we've been using the Plotly graphing library, which gives you more interactivity, in particular with this advanced hover mechanism. So for this, we use Sphinx Gallery for generating the gallery of examples, and it's possible to include examples using Plotly instead of Matplotlib, which is the default solution, by adding just a few lines of code. I'd like to finish with a few highlights of the two last releases, saying that we've been working on adding new algorithms. Here are just a few examples. We've been working on all kinds of things. A very common challenge in image processing is parameter selection, where you never exactly know which parameter you should choose for your algorithm. And therefore, we added one meta function in order to calibrate denoising estimators. And so this was one of the recent additions. We also have new registration algorithms, more clever superpixels algorithm, or rolling background subtraction, which was a very popular ImageJ plugin, and we now have it in scikit-image as well. In terms of trends, we have more and more 3D compatibility. It's not 100% 3D compatible, but all these functions have now an added 3D compatibility. And during the 2 last releases, we've worked a lot to extend our region props function, which is one of the most popular functions in scikit-image, in order to measure the properties of image labels. So now you can provide custom functions. It's compatible with multi-channel images. We have more properties. And we've also worked on performance, in particular in algorithmic improvements, like having more clever algorithms, more adapted to some cases, using lookup tables or single precision data, and also doing benchmarking in order to see which algorithm were taking a longer time than others. In terms of next steps, we are very excited that scikit-image got funded also by CSDI for EOSS round 3. And this time, the project, the grant will be led by Greg Lee, who recently moved to Quantsite. He will, of course, focus on growing the community and on maintenance, but focus in particular also on optimization, thanks to accelerator backends. And for the next steps, so scikit-image is a collection of algorithms, but we'd like to be used by a lot of people in interactive applications. And this is why we're excited that we used as backend for interactive workflows by packages such as Nafari, but also in Dash applications, which will be the subject of the next talk. Thank you very much, and I hope you have questions.
My name is Nick Christian. I'm the VP of Product and Head of Data Visualization at Plotly. Plotly, the company, who is also sort of the prime movers on the Plotly open source library and on Dash. So I worked very closely over the past year with Emma, and actually I'll be presenting this section myself just to ease on the sort of transitions, but I'll be mostly presenting work that Emma led and I worked on. So a little bit about Plotly and Dash. So the Plotly open source library is an interactive data visualization library. It's available for Python, R, and Julia, and it's got a JavaScript sort of backend that powers it all. And so the interactive visualization runs in web browsers. All of those libraries are MIT licensed, and the URL there is sort of the launching point for learning more about them. So we have at least 4 repos on the Plotly side, and then Dash is what we call a low-code analytical application framework, also available for Python, R, and Julia, and it enables folks to build web applications without writing any JavaScript. So it's also MIT licensed, and it has its own website. And so together, these 2 ecosystems are sort of quite tightly integrated and span a couple of dozen repos. You know, Dash is available in a number of languages, so is Plotly. There are a number of component libraries for Dash. So that's what we worked on over the past year with Emma. And I can't list everyone here, but a number of Plotly employees contributed to this work as well as a number of community members. So just to give a little flavor of the Plotly open source library, can be used to sort of make any of these charts. So a number of statistical graphics, scientific graphics, 3D graphics, polar charts, maps of various kinds, and sort of multidimensional visualization. So I'll spare you the blinking. And Dash can be used to build analytical web applications. And so probably the best thing I can do is I can show you this pre-recorded demo that was sort of built from our perspective, sort of out in the wild by a set of users prior to this CZI grant to sort of show you what folks were doing. So this is the work from a lab who built a tool called scikit. So what you're looking at here is an application that was entirely built in Python with no JavaScript, and it includes sort of typical scatter and bar charts. You can do selections. You can render sort of single channel and multi-channel images, and you can see that one graphic is sort of interacting with the other. So as you make selections on the one side, the other graphic is sort of evolving and responding to what's happening. So this is a pretty typical use case of an analytical application that someone in life sciences might want to build. Now, this particular application, is thousands of lines of code and is very difficult to build. And so what we wanted to do is to make it easier for folks to be able to build systems like this. Let me pause this image. Oops, it's looped. All right. Another example of a Dash application sort of in the wild is this one called in Delphi, which is an interface to a machine learning application intended for scientists using CRISPR. So this is in support of a publication that was published in Nature. The developers of this algorithm chose to use Dash to build an interface and put it on the public internet to their model to allow people to play with it and use it. And so one of the things that I love about this is that there's a little link at the bottom here as part of the app called, a shareable link to your results. And so this is a really good example of folks using Dash to build applications to support their research. So the CZI project that was funded as part of the Cycle 1 was entitled A Modular Suite of Advanced Bioimaging Tools with scikit-image and Dash. So Emma presented some of the work around scikit-image. The work that we did with the Plotly and Dash ecosystems, we basically framed it around the goal of making it easier for more life scientists to use Dash to support bioimaging work. So we knew that life scientists were using Dash already. The two examples I just showed are good examples of that. But the difficulty level to do what they did was quite high. And so we wanted to sort of lower the bar that people had to clear in order to be able to use Dash in their work, because we know that not everyone sort of is or wants to be a Python expert. And so we really wanted to make it as easy as possible for people to do this. And the approach that we adopted was to basically work sort of up and down the adoption pipeline. So of course, we added some new features to throughout the sort of Plotly and Dash ecosystem into scikit-image. We worked on performance, but we also worked on documentation, documentation content, documentation search, making it easier to find the documentation we already had, adding documentation for the new features, but also for the features we already had that were poorly documented. Going sort of a notch deeper, we used some of the CZI funds to directly engage with users on our very active community forum to understand sort of the docs that they were finding, the docs that they were not finding, directly helping people work with these technologies sort of to help them, but also to inform how we could better help members of the community in the future. And then beyond that, of course, we contributed conference talks, webinars, and podcasts to publicize our work. And sort of the central showcase of what we did with Plotly and Dash was to build a set of example apps. And so to give you a sense of sort of how that work progressed, here's sort of slightly generalized workflow of what building an example app looked like over the past year. So essentially, one of us, usually Emma, would sort of design a bioimaging app and start building it, and quite quickly sort of run aground and would not be able to figure out how to do it based on the documentation that we had. And so an immediate response to that was to improve the documentation so that not only we could move forward, but also people could follow in the trail that had been blazed. The next step was to discover that there's no easy way to do what we were trying to do in Dash. And so, well, we had to sort of do it the hard way. And then eventually, bumping into a missing feature. We just can't do what we're trying to do because Dash doesn't support it right now. And so the response to that was to work with the teams that maintain Dash and Plotly to add these features, notably around the display of multi-channel images and building systems for selecting and annotating features within images. A typical problem we ran into is that the end-to-end system was too slow, and so there was some performance work done to make it faster. And then when all was said and done, we could figure out how to do it. We did it the hard way. We added the features. We made them faster. It turned out that the result was too complicated, and we didn't think that your average sort of life scientist would be able to or willing to do this. So we sat down and sort of tried to figure out what are the generalizable pieces, what are the building blocks that would really comprise a modular suite of tools. And then we started building sort of reusable components to pack down that complexity either into the framework itself or into a component that folks could use to do their work. And so there are about 6 example applications that sort of went through this process over the past year and were written and rewritten and rewritten as new features were added, new faster ways of doing things were discovered and developed, and new reusable components were built. And the last few steps are pretty simple: to document the process, publish the sample apps, raise awareness of them by giving talks or posting about them on Twitter, writing a blog post, and then helping community members use these features directly. And so this is the sort of generalized process that we applied sort of over and over again to try and lower the difficulty bar so that folks could use Dash and Plotly and scikit-image to build the applications that would benefit their research. So getting to some sort of concrete examples of what we did, this time last year, Plotly did not have the capability, Plotly, the visualization library, did not have the capability of displaying full color images like this one. We could only display single channel images. And so we built an analog to Matplotlib's ImShow and spent a lot of time improving it, making it faster, making it work with facets, making it work with animation, which recently came out, recently meaning yesterday. As well as enabling users to be able to sort of annotate and select components of the image. We added some documentation to Dash. So this is a screenshot from the Dash documentation, specifically around image annotation with a number of executable examples. And both of these screenshots are from our documentation, which we worked on very hard over the past year, including building a new search system in each one, so one for Dash and one for Plotly, to make it easier for users to find what they're looking for, and really listening to the community telling us that our reference documentation was not good enough, was too slow, was too confusing. And so we've done a number of iterations on both the Dash and Plotly side to make features sort of easier to find. This is a screenshot from our community forum. We have a wonderful show and tell section. There's a link at the top of every page here. And this is where we encourage folks from the community to tell us what they've done with Dash. And you can see sort of the activity is pretty high: 5 days, 7 days, 8 days, 9 days. Basically every day we get to come to our forum and see a new example of what people have done. And so folks are building new Dash components, new Dash applications, new frameworks on top of Dash, and it's sort of very gratifying to see, and it creates a base of inspiration for other users. So you can see people saying, "Hey, like I'm really impressed with this. How did you do this? Like I've run into this problem myself," and so on and so forth. So that's been great, and we've spent a lot of time on the forum answering folks' questions to enable these show and tells to really sort of take shape. We have a gallery of Dash applications. So if you go to dash-gallery.plotly.host, these are the 11 applications that are tagged image, but we have about 90 different open source Dash applications on the website. At the moment, you can play with them directly, download the source. About half of them have equivalents written in Python and R, which is pretty exciting. And I'm going to dive into one specific application, which really kind of showcases all that's possible and all that's been done throughout the past year. So this is an application examining CT images of a COVID patient straight off our gallery server. So hopefully fast enough. All right. So here you can see it's essentially got 4 separate Plotly interactive figures. 2 of them are images and 2 of them are sort of standard information visualization. So here you can see a sort of top-down view, top-down slice, and then the other one is a sort of transverse slice. I can grab a slider and I can sort of scrub through, and you can see the indicator is moving on the other image. I can scrub through and find this gray region, which I'm told is an occlusion in the lung. And I can sort of try and scrub through over here and look for the same one. So you can see this sort of gray region over here represents the same occlusion. And I've got a kind of hard to read 3D mesh view down here. And so this has been generated with scikit-image. So one of the neat things you can do using the features that we've added over the past years is you can now select more or less precisely a specific region over here. And I'm going to select, do a rectangular selection over here and say, these represent the sort of same thing. And here you have a histogram of values of intensity levels. And so if I mouse over the occlusion, it's all around sort of the 150s. This part over here is very, very bright. So this spike here is the sort of white area that I've captured. So I'm going to further sub-select the data that I think is interesting. And then right now we've got an algorithm running in the background. It says updating. that's calculating and highlighting in red, the exact occlusion based on one lasso selection, one rectangular selection, and then one selection in essentially intensity space, and then viewing the results as an overlay here, and I can sort of scrub around and see different slices. So it's not perfect. It's done a reasonable job, but this app really shows what you can do with sort of Dash, Plotly, and scikit-image. And I'm very proud to say on behalf of the team that it's now possible to write this app in 400 lines of Python code because we've generalized sort of this slicer component and generally sort of try to pack as much of the complexity away so that folks don't really have to think about web traffic and high performance rendering, and they can mostly focus on expressing the user interaction and the algorithm that they want to express. So this is one of the apps that that sort of went through this process of simplifying and refining and helping us find the pitfalls and problems with the Dash and Plotly ecosystem. Back to my presentation, I've got one more little app which I think is kind of neat. This is interactive machine learning for image segmentation. So I've got another image here. I've got a little palette of colors I can use, so I can pick up a color and I can basically scribble on this image. I want the classifier to make this all orange. And in fact, I made a mistake. I don't think I want that little area. There you go. I want the classifier to make this whole area here blue. This little net looking thing I want to be green. And this sort of squiggle I would like to be yellow. And then I can basically run the machine learning model in real time, show the segmentation. And you can see that it's done a reasonable job. It sort of colored this area in orange. I should have chosen something higher contrast, but it did an okay job for the squiggly bit here in yellow. The networky bit here got made in green. And so this whole app again is the kind of thing that you can build with just a couple hundred lines of Python code, but it really integrates the user interface aspect, the image processing aspect, and the image display aspect, as well as whatever algorithm is under use. And so in this case, you can either download the classified image or download the classifier. So neither of these apps are necessarily the kind of thing that you would directly be using, but we chose them as sort of representative of the sample of the space of apps and techniques that life scientists might want to use to build applications that they either use for themselves in their labs, put online for sort of distributed sort of crowdsourced data collection, or much like the Indelphi example, use an app like this to present the results of their work, either giving access to a data set, letting people directly use an algorithm so as to better understand what it's doing. These are really sort of foundational technologies that we are putting out there in the world and hoping that people will use to build on top of. This is the sort of final slide I've got is a screenshot from the blog that we ran sort of sporadically throughout the past year, posting updates on sort of what we've done throughout the year that's been enabled by this grant. So we thank the CZI profoundly for enabling us to do this, because we've done some really interesting work. And I would recommend to anyone who's entering cycle 3, it certainly makes it a lot easier to sort of look back on what you've done if you keep a blog and keep it public. So we probably should have started earlier and done it a bit more often. So I'd recommend that to anyone starting now. And that's all I've got in terms of presentation. So thanks very much. I'm happy to take questions and leave room for Emma to take questions as well.
Well, one question, just kind of reflecting back on Eric's question from last time. You may have covered this a little bit, but how do you support the open source financially? What's your way? Is it all through grants? Are there support, or like how do you cover all this development? I mean, it's pretty amazing stuff you guys have done.
Is that question aimed at Emma or myself, or just generally? Or
it seems like you kind of interconnected, related projects. So for both of you.
Yeah, go ahead, Emma.
So in terms for scikit-image, live funding was the first time scikit-image received funding. So before the EOSS program, Núñez Iglesias had already been funded as an imaging fellow. So CZI had several programs, and so we have benefited from EOSS, but also from this other program. And Berkeley, in particular BIDS, has also contributed some funding, but most of the development is done by volunteers. So it's really a small fraction which is funded by organizations, and also people do it mostly during their free time. Yeah.
On the other hand, we've got Plotly and Dash are, I would say, almost entirely funded by Plotly, the company. And we have kind of a unique model in a way. So we do sell commercial products, which I won't talk about here, but sort of hover around Dash. We also have a very large number of open source users who basically will phone us up and say, hey, look, we love your visualization library, but it doesn't, you know, do X, Y, Z, the last sort of 10% of the features that we need. Can we pay you to add them, please? And we do. And so we do take direct sponsorship from companies. And I think that the fact that we have sort of a successful software company running in the background helps us smooth over the sort of peaks and troughs there, because sometimes there's long troughs and sometimes there's intense peaks. And so we are basically, you know, a commercial software company that runs a successful open source project. And we are happy to accept contributions, and we get a bunch. And we also put a lot of staff time into the open source projects because our commercial customers also use our open source projects. So we don't sell them a different set of software. We sell them additional software on top of our open core. So I won't say it's like the easiest model to run, but we mostly succeed.
Question?
Please go ahead.
It was great. I'm an imaging guy, and so and I'm also a Plotly user. I stumbled into Plotly recently for a project, and I really like what you guys have done and have used it a lot for our own work. Question about what kind of images you're handling on the back end. So are those all 8-bit images? Are those RGBs? What are those formats? This kind of stuff that we think about. It'd be great to talk about that maybe offline. But you know, what kind of imaging data would you support?
Go ahead, Emma.
Should I start, Nick? Maybe?
Yeah, go ahead, please.
So in scikit-image, you can handle many kinds of images as long as they are in the form of NumPy arrays. So they can be either floating points array, or 8 bits, or 16 bits. So most algorithm will accept all kinds, sometimes internally converting to another type. So that's for the data types which we accept. In terms of input and output, we delegate to third-party libraries such as ImageIO to read a large variety of image files. I hope this answers your question. Otherwise, I'd be happy to be in touch. It's exciting if you say that you've been using Plotly and image processing.
Yeah, so basically anything I can put in NumPy array is what.
Yeah.
Okay, that's fine.